This video is Chapter 8 Notes, Part 3, all about meiosis. Meiosis is used in sexual reproduction. There are only a few cells in your body that can undergo meiosis. So it's not as though a skin cell could create an egg cell. So you have cells in your testes or cells in your ovaries that will then make egg and sperm via a process called meiosis. Meiosis has some similarities to mitosis, but also some key differences. During mitosis, we begin with one cell and end with two cells that are clones of each other and are identical to the original starting cell. In meiosis, on the other hand, we are creating four cells. All of those cells are genetically different from each other and genetically different from the parent cell. During meiosis, the original cell contains a full set of chromosomes, whereas the resulting cells, which will become egg or sperm, will contain only half the amount of the DNA. We call those resulting cells haploid. A haploid cell is a sex cell, and it is a sex cell that contains half the chromosomes. So a normal human has 46 chromosomes. They would pass on 23 to their offspring. That offspring would then get the other 23 from the other parent to make a little baby who has the normal amount required. So a haploid cell is an egg or sperm cell that contains half the amount of chromosomes. Di means two. A diploid cell contains a full set of chromosomes from two parents, meaning there's two copies of every chromosome, a copy from mom and a copy from dad, whereas haploid cells do not contain as much DNA. The word zygote is a fertilized egg. A fertilized egg is a diploid cell. You can see shown here in the picture, there is an egg cell. An egg cell is huge in comparison to sperm. Essentially, sperm is just DNA that's swimming. There's not a lot else in the cell, whereas an egg cell will contain food storage and a bunch of organelles that the cell will need to function normally. So we've got an egg cell here on the left and a sperm cell. As soon as that sperm cell shoots its DNA into that egg cell, I would call that a zygote or a fertilized egg. Homologous chromosomes are chromosomes that have the same genes, but maybe different alleles. So us as humans have 46 chromosomes, but they exist as 23 homologous pairs. So shown here are all 46 of those chromosomes, but they are paired with those that are homologous to one another, resulting in 23 pairs. These sex chromosomes are considered a pair. In each pair of homologous chromosomes, we see the same traits, meaning the same genes, but maybe different versions, aka different alleles of that gene. So, for example, this is an oversimplified example, but let's say if we're talking about eye color would exist on both of the chromosome number 1. They are homologous, the gene is eye color, but maybe one codes for blue eyes, I got that from mom, and one codes from brown eyes, I got that from dad. So I would call those then homologous because they are similar but not identical. you've got about 25,000 genes in your body. So you've got 25,000 copies from mom and 25,000 copies from dad. So we have the same genes, but they might be different versions. For a lot of the genes that make you a human, well, your mom and dad are also humans. So those genes would be perfect photocopies of each other. They would be the same allele at those genes that make you a human. But what makes you have certain physical traits or chemical traits in your body might be inherited. You might have inherited different copies from your mom versus your dad. So this kind of says the exact same thing. So crossing over involves those homologous chromosomes, and crossing over helps to explain why there's so much variation in sexually reproducing organisms. And in crossing over, essentially what happens is during prophase, the homologous pairs, let's say I got this white one from mom and I got this darker one from dad, and you can see that I've got capital letters when we talk about like big A, big B, little b, when we talk about doing like Punnett squares and good old-fashioned genetics. I've got capital letters from mom and lowercase letters from dad. So if these chromosomes line up and crossing over does not occur, I might potentially inherit this entire chromosome from my mom, or I could get this entire one from dad. But actually what happens during crossing over is there's little enzymes that snip the chromosomes and re-glue them together into a pattern that did not exist in either original parent. So here I have combinations of capital with lowercase. I never had that to begin with, capital with lowercase. And lowercase with capital, I never had that to begin with either. So crossing over creates new recombinants, a recombinant being a combination of genes that wasn't there to begin with. So I create new recombinants that provide endless options of what the egg and sperm could look like. Because remember, if there's 500 or 1,000 genes on here, the amount of places it could be cut is huge. And I have these little Verizon guys on here because crossing over kind of reminds me of them. As they inflate and deflate the chromosomes, if you imagine these are chromosomes that are moving around during prophase, as they touch pieces, so like as these Verizon guys crash into each other, there would be little enzymes there in prophase that would snip them apart and re-glue them into a new and different combination. So crossing over hugely increases genetic variation. It's more ways that I can get chromosomes that were not there to begin with. This color-coded picture on the left shows that pretty well. I've got a blue chromosome and a red chromosome. I can create offspring that do not get either blue or red, but rather get a little combination of each. And those are my recombinant chromosomes. In crossing over, the farther two genes are apart, the more likely crossing over will occur. So the chance of A and B getting snipped, like the chance of that chromosome getting snipped between A and B, is not as big as the chance of getting snipped between D and E, simply because if two things are far apart, there's more space for the machinery to get in there and do the cutting and re-gluing of those chromosomes. So some traits tend to always be inherited together, and the reason for that is just because they are physically so close together on the chromosome that they always seem to travel and get inherited together. So crossing over explains how we can create chromosomes that are different from either of the parent chromosomes. Independent assortment is another explanation for how we can end up with offspring that is so different from each other and also so different from the parent. During independent assortment, we see that chromosomes line up randomly. If you imagine that you have in your ovary or in your testes where you're going to create a sex cell, you're going to pass on 23 of your chromosomes. You could pass on 1 from mom and 22 from dad. You could pass on 2 from mom and 21 from dad. You could pass on 3 from mom and 20 from dad, and so on. And then also you could pass on let's say you're passing on only 1 from mom. It could be chromosome number 1. It could be chromosome number 2. It could be chromosome number 3. It could be chromosome number up to 23. So there's a huge amount of combinations that can be produced from 23 things lining up in the center of the cell. Shown here in the diagram is 2 separate homologous pairs. So I have a big pair and a small pair, and a big pair and a small pair. So I'm imagining this is 2 separate homologous chromosomes. So I've got chromosome number 1 and chromosome number 2, chromosome number 1 and chromosome number 2. Well, in my sex cell, I'm going to pass on half of it. So there are multiple different combinations that can be formed. I could have both blues going to the left. I could have 1 blue and 1 red going to the left. You can imagine I could have many possibilities if in reality I have 23 of these pairs. So this just illustrates that there's a huge amount of combinations that can be produced. The chance of you making the same egg cell twice or the same sperm cell twice is really not likely, especially when we add in the fact that crossing over can occur, and in reality these would be pieces of each. So there's a huge chance of genetic variation due to crossing over and independent assortment. So kind of thinking big picture, back to the big picture here, the goal of meiosis is to create haploid sex cells that contain half the amount of DNA. The majority of the cells in your body are not doing meiosis. Meiosis occurs when one cell replicates its DNA. So, oh yes, still interphase is going to happen, and photocopying, replicating DNA will occur. And then there are 2 divisions. So if you think about a human cell would contain 46 chromosomes, it would get replicated to 92. That number gets split in half twice, creating egg and sperm that contains half the amount of DNA as the starting cell. So there are 2 rounds of division in meiosis where the cell essentially goes through the steps of mitosis twice. There's obviously some differences because it's not a cloning process. So we call those 2 steps meiosis 1 and meiosis 2. In meiosis 1, we still have interphase. The cell is going to wait for a signal to divide. It's going to get bigger. It's going to replicate its DNA. It's going to replicate the organelles. Then, during prophase, my homologous chromosomes pair up. Also, my nuclear membrane starts to degrade. During metaphase, I have the DNA lined up at the equator of the cell. During anaphase, my homologous pairs get separated. The cell then reforms 2 nuclei, pinches in 2, and I have 2 cells at the conclusion of meiosis 1. These cells are not used in reproduction yet because they don't contain half the DNA yet. So the process continues in the form of meiosis 2. Meiosis II is essentially the same as mitosis. But now in meiosis II, we have 2 cells that we're starting with because we just ended with 2 in the previous meiosis I. Each of those cells will then undergo prophase. There is no DNA replication before meiosis II, so that interphase does look different. During prophase II, nuclear membrane dissolves. Metaphase, DNA lines up at the center. Sister chromatids separate during anaphase. Nuclear membrane reforms during telophase. And at the conclusion of cytokinesis, I then have 4 cells that are genetically different from each other and contain only half the DNA as the starting cell. This then shows exactly that. During interphase, chromosomes duplicate, homologous chromosomes pair up, line up at the center, homologous chromosomes separate. Cell pinches into 2, 2 cells are formed. Then we have those similar steps repeated. There is no interphase where DNA gets replicated again, but we have prophase occurring again where the nuclear membrane dissolves, DNA lines up at the center, sister chromatids separate, and then we have haploid cells that are formed at the end. So to conclude, there's some slides that show similarities and differences. Major differences: meiosis occurs with 2 divisions. It's creating haploid cells. It's creating 4 cells instead of 2. And then independent assortment and crossing over occur to give even more possibilities of sex cells that can be formed. I would read through these.